Our next speaker is Eugene Koonin, a senior investigator at the NCBI, the NLM, and the NIH. So Eugene, if you're there, please go ahead. Thank you.
Yes, thank you very much for the introduction. Let me start sharing my screen. Good morning, good afternoon. Thank you very much, Pierre, for the invitation to speak at this truly fascinating meeting. I'm pleased to be here. And in this talk, I'll present you some observations and ideas on the relationship between primordial replicators and origin of viruses and cells. So we shall move from the fascinating world of subterranean chemistry and microbiology to the more rarefied areas of theoretical biology and comparative genomics. But first of all, what I want to mention here, I hope in the spirit of the sort of planetary scale of this meeting, is that the most abundant, the most common, the most numerous biological entities on Earth are viruses. We have just heard about the interesting subterranean regions of the biosphere where one has to look into biological needle in a abiotic haystack, but on many habitats on this planet, it is indeed the opposite. They are looking into a huge biological haystack, and the main components of that haystack are viruses. You can find hyper-astronomical numbers of virus particles in a variety of habitats, up to 1 billion virus particles per 1 cubic centimeter of, say, sea water in rich habitats, which is very often an order up to two orders of magnitude more than you find cells of any kind in the same habitats. And viruses are also the huge genetic reservoirs in the biosphere, so that the majority of distinct genes are actually in the virus genomes. This translates into hyper astronomic numbers of viruses on the planetary scale. And just for the sort of fun comparison, at any given moment, the best estimate is about 10 to the 31st virions on Earth, compared to about 10 to the 24th stars in the observable universe. Viruses, however, are notable not only for their abundance, but for something that may be more fundamental, for several features that may be more fundamental. To begin with, it's textbook knowledge, but I think it's important to keep in mind and recall when we think about evolution of life, especially its early stages. Viruses encompass all possible types of strategies of genome replication and expression. Now, we all cellular life forms employ the same scheme of genetic information storage and expression. From double-stranded DNA to various, by transcription to various RNA molecules, and from messenger RNA to proteins by translation. This is what happens in every cell on Earth. But what happens with viruses is very different. We know that virus genomes, in the case of viruses, the genome is the nucleic acid incorporated into the virion. And virus genomes can be represented by all thinkable forms of nucleic acid, single-stranded RNAs of both polarities, double-stranded RNAs, single-stranded RNAs or double-stranded DNA followed by reverse transcription, and single-stranded DNA, all possible forms of nucleic acids and all possible strategies of genome replication and expression, which leads to a provocative idea, which I will discuss a bit later in this talk, that virus, perhaps not viruses per se, but virus-like entities might have been nature's original genetic test systems that had been tried by early evolution for storage and expression of genetic information. This is buttressed by the ubiquity and inevitability of the existence of genetic parasites, which I'll also discuss in a moment. Ubiquity means that effectively all cellular life forms, with possible exceptions of some intracellular parasitic and symbiotic bacteria, are infected by viruses, or at least afflicted by some genetic parasites. And inevitability means that actually it seems to be theoretically inevitable that genetic parasites emerge in any replicator system. And finally, by way of introduction, I want to note that evolution of viruses is a complex matter, in the sense that there is no common ancestry to all viruses, not a single universal gene. So in a sense, this is a much higher complexity than the entire evolution of cellular life forms. So to begin with, I want to discuss what I consider to be a theoretical argument for inevitability of genetic parasites. Namely that parasites or genetic parasites or cheaters in terms of game theory evolve in any replicator system at all. Speaking in sort of a cynical manner, as soon as there is something to steal, a resource such as replicase, for instance, someone in the course of evolution will find a way to live by stealing. More formally and more seriously, however, I would argue that parasite protected states of replicators are low entropy states that therefore are thermodynamically unstable. So in order to make this argument, we consider the very simplest replicator system that is a nucleic acid, a replicator that encodes a replicase, which catalyzes the replication of this very genome or other, and can also function in trans, catalyze replication of other genomes, and that depends on the feedback from the replication errors, mutations. So the entire system is capable of evolution by selection and genetic drift. Incidentally, this is not a theoretical construction. This is exactly how the simplest RNA viruses are built up. So in such a replicator, there are two types of signals, two types of information. One that we will call the replication expression signal, and that is a coding sequence for the replicase. And the other type of signals is what I will call replication recognition symbol, and this is something much simpler. This is simply the sequences that need to be recognized by the replicase in order to replicate a nucleic acid. So this is marked here in red schematically. This is relatively simple signal, whereas what is marked in blue is the replicase expression signal, which is far more complicated. Given these two types of signals, it is clear that the full-fledged replicator that is that both encodes a replicase and is replicated by that replicase. Is a low entropy state. Simply put, there are very few states that correspond to a fully functional replicator. In contrast, there are a great many ways in which you can impair that replication expression signal and make a parasite that retains the replication recognition signal. And to that situation, we can directly apply the famous Watson formula and come up with the understanding that the parasite state are states with much higher entropy than the autonomous replicator state. And therefore, the parasite-free state is inherently unstable. The emergence of parasites is inevitable. And furthermore, in evolving biological replicator parasite systems, complexity increases as a result of frustration, that is, competing objectives in different components. Minimization of parasite genome versus preservation of all the information that is necessary also for the parasites in the fully fledged replicator. Incidentally, this is not all a theoretical construct, but it is a theoretical scheme that fully recapitulates the famous experiments in Saul Spiegelman's lab reported by now more than 50 years ago, whereby through serial transfers of the replicating RNA molecule, that of bacteriophage Q beta, between test tubes containing all the necessary materials for building blocks for replication and the excess of phage replicase, through these serial transfers, the RNA replicons shrunk to 10% of the original size, exactly losing all the replication expression systems, just as illustrated in those theoretical schemes. So I think this model gives us a strong argument for the inevitable emergence of genetic parasites in any replicator system. We will now leave these theoretical arguments and talk in a more qualitative way, but also using a plethora of available data on the subject that is very dear and exciting to me, namely origin of viruses. So over the last century, more or less since the discovery of bacteriophages by Derrell more than 100 years ago, three groups of evolutionary scenarios for the origin of viruses have been discussed. To begin, so to speak, from the very beginning, there were virus early hypotheses, to which I already alluded to in the very beginning of my talk, and according to which viruses are direct descendants of the replicons that comprised the primordial pool of replicators at the precellular stage of evolution. In the early days, the terminology might have been different, but the ideas were the same. So basically, viruses, or at least according to these types of scenarios, viruses, or at least virus-like replicators, precede the cellular stage of evolution. The opposite of this is the group of regression hypothesis, according to which viruses evolve by degradative evolution of the simplest parasitic cells, such as, for instance, similar to the known intracellular parasitic and symbiotic bacteria. Viruses are considered the ultimate stage of this regressive evolution. And finally, probably the most popular in the pre-genomic era, escaped gene hypothesis, according to which viruses are descendants of, so to speak, regular cellular genes that have gained replicative autonomy. All these scenarios, however interesting they might be, have been more or less a speculative subject for decades. But the situation changed in the last 2 or 3 decades with the advances of genomics and metagenomics. Now we have many thousands of genomic sequences of all kinds of viruses and other mobile elements from all kinds of diverse families and higher taxa, and by comparing the so-called virus hallmark genes, that is, genes that encode the key components of the virus replication machinery and structural build-up, can make conclusions on the evolution and origin, on deep evolution and origin of viruses. As I mentioned, there isn't a single universal gene among viruses, making things more complicated and interesting, but fortunately, there are these highly conserved hallmark genes that are shared by a large group of viruses, and in particular by different so-called Baltimore classes, that is, classes of viruses that employ different strategies of different nucleic acids and genomes, and different strategies of genome replication and expression. These are, just to give you examples, reverse transcriptase, RNA-dependent RNA polymerase, superfamily tree helicase, rolling circle replication, initiation, and the nuclease, single jelly roll capsid proteins, double jelly roll capsid proteins comprising icosahedral virions, and a few other highly conserved hallmark proteins. By exploring by sequence and structure comparison the evolutionary histories of these genes, we can make conclusions on evolution and even origin of viruses. So to begin with the virus replication machinery. So the majority of viruses encode at least one key enzyme that catalyzes the replication of the virus genome, such as reverse transcriptase, RNA-dependent RNA polymerase, DNA polymerase B, that is responsible for the replication of most of the DNA viruses, DNA polymerase A, which is responsible for replication of some of viruses, primase, or initiation and the nuclease that are also found in large group of viruses. These are highly diverse proteins. Within each of these rectangles here, there is some sequence similarity that can be detected by, say, iterative PSI blast searches. But between these rectangles, you really cannot see. However, they're highly diverse proteins, which is exacerbated by the fast evolution characteristic of viruses. However, then you switch to structural comparisons. You detect remarkable unity behind all this variety of virus replication enzymes. Basically, they are all, each of these proteins has its core fault, the so-called RRM, RNA recognition motif domain. It's very simple arrangements of the sheet and a couple of alpha helices illustrated here. This is one of the most common nucleic acid binding domains in nature, and the majority of the forms of this domain are actually not enzymatically active. They just bind RNA or DNA molecules and facilitate various processes, splicing, and many others. But on a variety of occasions, these binding domains evolved catalytic centers and become highly efficient polymerases. So from these observations, our conclusion is that virus replication machineries actually do come from, in all likelihood, do come from the primordial pool of replicators. More precisely, even come from the interface of the primordial RNA world and the more modern biological system, where such domains might have been cofactors of ribozyme polymerases. If we now move to the structural modules of viruses, we see a very different picture. What we see here is that for the increasing, growing majority of capsid proteins and other major structural components of virions, we can identify very specific ancestors among cellular proteins. Fittingly, the majority of these inferred ancestors are carbohydrate or nucleic acid binding proteins. This is not just shared core domains, but specific ancestry from distinct hosts. Putting together these observations on the apparent origins of the virus replication machinery and the repertoire of virus structural proteins, we come up with this hybrid scenario of virus evolution, which is a combination of the, coming back to the more conceptual slide that I showed a few minutes ago, a combination of the virus early and escape gene scenarios, whereby the replicative part of the viral of the virus genetic repertoire do come from the ancestral pool of replicators, whereas the structural components represent escape or rather scavenged genes that have been recruited from hosts at different stages on evolution on many independent occasions, some very early in the evolution of life, as we shall discuss in a moment, others later. So basically, the world of viruses can be schematically represented by this type of network where the major monophyletic divisions known as realms of viruses are superimposed over the Baltimore classes, the classes of viruses with different ways or with different nucleic acids used in genomes and different ways of genome replication and expression, whereby all these groups are linked together by virus hallmark genes. I would like to next, in order to get a better insight into the origin and evolution of the world of viruses, to consider the last universal cellular ancestor and its virome that we can reconstruct from the diversity of extant viruses, and that appears to have been quite a complex virus, quite a complex collection of viruses that already infected that last universal cellular ancestor. So on this slide, with Mark Kropovich, we combine all the available information on the representation of different major groups of viruses among the bacterial phyla. I wouldn't go into the fine details of this, but you can see that in quite many of the bacterial phyla, a great diversity of viruses, different viruses, are represented. And to the very best of my understanding, where we do not have viruses, this is not because they don't exist. This is just because, regrettably, so far they have not been studied. But this is being failed quickly. And so we can have a similar look at archaea and notice something remarkable, namely that although there are some overlaps in the form of these tailed viruses, tailed bacteriophages and archaeoviruses, and viruses like these, viruses with icosahedral capsids, most of the diversity of archaeoviruses are actually distinct and different, not represented in bacteria. So from these comparisons, we can perform a parsimony reconstruction of the virome of LITU. And what we can see here is that although the diversity of that virome was, in all likelihood, considerably less than the diversity of modern bacterial or archaeal viromes, it still was already quite a complex virome with multiple groups within the two major realms of double-stranded DNA viruses already represented there, already infecting the last universal cellular ancestors. It may seem somewhat paradoxical that this ancient virome was apparently dominated by double-stranded RNA virus, double-stranded DNA viruses, even if it was not that long on geological scale after the hypothetical primordial RNA world, but this is where the observation point with the RNA viruses being subsequent derivations not inherited, or at least mostly not inherited from the primordial pool. Moreover, what we can trace by sequence and primarily structural comparison of virus capsid proteins is a complex history of virus evolution prior to LITU. So there are two major types of the capsid proteins in double-stranded DNA viruses. The spheric icosahedral viruses have the so-called jelly roll capsid proteins, for which we were able to identify cellular ancestors and which have been recruited by viruses at some very early stage of virus evolution. And then prior to LITU, also very early on, what happened during virus evolution was the duplication of the single jelly roll proteins and the fusion of the duplicated copies into the double jelly roll capsid protein, which is the main capsid protein in the great variety of double-stranded DNA viruses. And the other major realm of double-stranded DNA viruses is these tailed viruses, which use the so-called HK97 major capsid proteins, completely unrelated to this one, which also has its own complex evolutionary history, where one protein inserted in the middle of another. So there has been quite complex evolutionary history of viruses and major and hallmark virus proteins prior to LITU, which implies high complexity of LITU itself. I tend to think now that think of LITU now not as a single population of a single species, but a community of diverse organisms. And to finish my talk, I'll just lead you from some more speculative conceptual considerations on the very beginnings of life and the very beginnings of genetic parasites. How could it all have started? Could there have been viruses before cells and replicators before reproducers. We shall discuss these definitions in a moment. But first, I want to quote this famous classic essay by J.B.S. Haldane, great 20th century scientist, one of the founders of population genetics, who was thinking deeply, one of the first, about evolution of life. And he wrote that life may have remained in the virus stage for many millions of years before a suitable assemblage of elementary units was brought together in the first cell. So this was published back in 1928, where no one had a good idea of what viruses actually were, but Haldane was famous for these types of insights. Nowadays, however, there is a different train of thinking, and this is a quote from a much later, but also a famous paper by David Moreira et Puri Lopez Garcia, saying, here we contend that there is strong evidence against the notion that viruses are alive and represent ancient lineages of the tree of life. In other words, these authors believe that Haldane's idea was wrong. So let's spend a moment talking about replicators and reproducers and where viruses belong in this scheme. So viruses are a special kind of replicate. Biological entities that replicate their genomes and encode some means, like we talked about replicases and the others, encoding some means for replicating their genomes, but lacking metabolism and in particular energy transformation. And viruses are a distinct variety of replicators that encode their own capsid proteins, that encode proteins that encases the genome into a virion. In this recent paper on virus definition, my colleagues and I presented a conceptual scheme of the relationship within the replicator space, placing here what we call the orthovirosphere, typical viruses with its 4 major realms and a few smaller ones surrounded by what we call the peri-virosphere, which is inhabited by various virus-like replicators, derivatives of viruses, apparent derivatives of viruses that have lost capsid proteins, or perhaps ancestors of viruses that have not acquired capsid proteins just yet. And all this is embedded into the wider replicator space, which is inhabited by transposons, plasmids, all kinds of replicators. Basically, replicators span the plane that is bounded by selfishness/cooperativity and mobility/replicative autonomy, starting with fully cooperative and immobile replicators, which are cellular genomes and going all the way through all sorts of semi-independent or independent replicators to the aggressive lytic viruses. All kinds of strategies are implemented by replicators. And so, after this introduction into replicators, I want to discuss a conceptual scenario for the origin of life that we have been lately thinking about. It's still unpublished material. We have been thinking about and discussing this a lot with my colleague Puri Lopez Garcia from Université Paris-Saclay, on the origin of cooperation and conflict and the origin of life in this context, what we call a mutualistic scenario, which involves 2 principal types of propagating biological entities, reproducers where physical structure reproduces and genome replication is insufficient to reproduce this structure, so omnicellular cells and replicators, various kinds of genetic elements, which we have already discussed. So according to their thinking, origin of life is a mutualistic merger of reproducers and replicators. So this scenario would start with what I would call pre-life, which is proto-metabolic networks of autocatalytic reactions compartmentalized within vesicles or other types of compartments. Importantly, selection occurs already at this stage, what may be called survival of the most persistent, at the stage of evolution of pure replicators. Conceivably at this stage, nucleotides and possibly amino acids serve as cofactors and were driving forces for nucleotide and amino acid accumulation. Now, upon accumulation of nucleotides, the polymerization would be enabled and ribozyme catalyzed evolve, including amino acylating ribozyme, which we know from chemical experiments emerge quite early. So to speak, the real-life stage. But we still have here evolution of pure reproducers for stability and persistence. The next stage would be origin of replicators, a difficult stage, admittedly, with most likely ribozyme polymerases, where sequences of highly efficient ribozymes would be memorized and selection for replication would start. This is what I would call a proto-life stage, where memory of the preceding states of the system is stored in RNA sequence. And finally, the origin of what deserves to be called life occurs where when reproducer components, not only the replicators themselves, but the components of reproducers, become encoded by replicators. Under this scenario, the origin of life is a merger of reproducers and replicators, whereby replicator-containing entities outcompete reproducers lacking replicators. And as I already referred to, under this scenario, a very early stage in the evolution of life is split of replicators into cooperators, cellular genomes and some symbionts, and parasites, which is concomitant basically with the origin of efficient replication. I will stop here. I think this, I didn't include the conclusion slides, but I think this scenario, despite its conceptual nature, is already amenable to experimental testing. I want to mention the contributors, my colleagues, key participants, Misha Kastelsson from Radboud University in the Netherlands, and Yuri Wolf, who are involved in a variety of theoretical studies and physical models of early evolution. Puri Lopez Garcia from Université Paris-Saclay, with whom we developed these conceptual models, Mark Kropovich and Valerian Dola, Institut Pasteur and Oregon State University, who are my main collaborators into the more specific issues of the origin and evolution of viruses. Thank you very much for your attention. I will be delighted to answer any questions if there are some.
Wonderful. Thank you so much, Eugene. It's always great to listen to your talks. Thank you so much. While people are posting, maybe people are posting some questions, I did see that there was a discussion around whether they're more viruses or more stars, but I suppose that goes down to the numbers. What I was going to ask you is the merger of these ideas of reproduces, sorry, for some reason, I don't know why you can't really see me that well, but anyway, the merger of the reproducers and the replicators, it strikes me as there's an analogy there with Ganti's chemoton. So Ganti's chemoton really fits in, brings these two pieces together. And I wonder if you would agree with me that it does resonate with what
not only I agree with you, Pierre, but Ganti's chemoton was really one of the main stimuli for developing these ideas. So absolutely.
Okay, thank you. And then I wanted to ask you as well, I was wondering, the issue around Morera, the philosophical discussion, I suppose it really comes down to how one defines life. And there are all these different definitions of what life is. But I was wondering why Morera excluded viruses from the tree of life. Was there any particular, I suppose, because they're not autonomous, or they're not autonomous?
No, the issue that that paper was more complicated and was more partly philosophical, as you said, but partly quite empirical. This was stimulated by the discovery of giant viruses by Claverie and Roll, and the attempts to build a phylogenetic tree using the sequences of amino acid tRNA synthetases encoded by giant viruses, suggesting that these giant viruses comprise something like a fourth domain of life. But these were really somewhat naive attempts at reconstruction, badly affected by long branch attraction artifacts. And when we did it more carefully, it's very clear that these genes, the homologs of cellular universal genes encoded by viruses, have been acquired at different stages of evolution. And in that sense, they cannot and should not be included in the tree of life at the same level as the cellular homologs. As for the philosophy, yes, this comes down to how life is defined, but I think it is very important for, not philosophically, but practically, for in order to work on theory and experiment, to keep in mind that however important replication genetic material might be, it never occurs in a vacuum. It needs to occur in compartments where all the necessary materials are provided.
So that would suggest to me as well that, new viruses are emerging all the time, out of whether it's out of complex organisms or whether it's out of unicellular organisms. There will be parasitic elements that every now and then, bud out of the original host. So new viruses, I would imagine, are emerging all the time.
All the time might be a bit of an exaggeration. That it's still not a completely trivial task to make a virus, but it is true. I think it's now undeniably true that different groups of viruses occurs on tens of occasions, not hundreds, during the evolution of life. We have traced many specific cases.
Okay. Excellent, thank you. It's looking in the Q&A here. Given that Luca's virome was primarily double-stranded DNA, do you propose RNA viruses came from a novel event or evolved from double-stranded DNA?
Okay. No, there is no indication of RNA viruses evolving from double-stranded DNA viruses. I think this can be effectively ruled out. Moreover, the replicative module, replicative enzymes of RNA viruses are actually very simple compact polymerases that in all likelihood do come from the primordial pool of replicators. But the origin of viruses per se, or of RNA viruses per se, apparently occurred already after the divergence of bacteria and archaea for the first time in the bacterial domain of life.